Good afternoon everyone. Good afternoon, Shafia. Okay. Okay, what about others? Okay, so today we'll finish the transposons as well as we'll solve the questions from the transposons which were asked in the previous years. Okay. Okay. Okay, no problem. Let's continue. Let others join. Okay, so in the yesterday last class, friends, we were discussing about the transposable elements. If every one of you quickly recall my words, remember everyone, if you quickly recall my words, we have discussed first different type of DNA sequences. Why did I tell you to remember the different type of DNA sequences, friends? Because our transposable elements. Good afternoon, Priya. Continue. Good afternoon, Shivani. Our transposable elements basically comes under the category of moderately repetitive DNA. Good afternoon, Deepika. In order to understand this, friends, we have started studying about the different type of DNA sequences. Later, we have understand the characteristic of the transposable elements. I hope, friends, every one of you remember this particular concept especially. Good afternoon, Vinita. I have clearly told to all of you, friends, the terminal inverted repeats. Friends, please listen my statements very carefully. The terminal inverted repeats, these are the integral parts of the transposable element. That is what I told to you. And what I also told to you, friends, the flanking direct repeats, everyone listen, the flanking direct repeats are not the integral parts of the transposable element. I have clearly told to you that, friends, whenever the transposable element is moving from one place to the another place, I told you that the terminal inverted repeats also will move. There is no doubt about it. But do you think the flanking direct repeats will move along with the transposon? Never ever. Please don't get confused with that. And friends, we started studying about the type of transposable elements. I hope every student remember. I'll quickly summarize in 2 minutes. We have studied about the RNA transposons, as well as, friends, we have studied about the DNA transposon. And good afternoon, Saima. Continue. What is the major statement I have told to all of you? I have told you that in case of humans, friends, everyone listen. In case of human, DNA transposons are present as well as the RNA transposons also present. But what I said to you, friends, RNA transposons are more common in case of humans. Do not confuse with this, friends. If you look into the prokaryotic, like, friends, bacteria, what did I tell you? Majorly DNA transposable elements are present. Please kindly do not get confused with that particular point. And friends, while studying the DNA transposable element, I hope every student remember the DNA transposable element basically follows the two methods. What is that, friends? One is the replicative DNA transposition, and another one is what, friends? Non-replicative DNA transposition. I have already told to all of you, friends, to remember the most important point, that is, in the replicative DNA transposons, everyone remember, always copy and paste mechanism can be shown. But friends, in case of a non-replicative DNA transposon, what I want you people to remember, friends, I want you to remember that it will always show, friends, cut and paste mechanisms only. Please don't get confused with this particular point. And what's the another statement you have to remember? In case of a replicative DNA transposons, friends, everyone please remember, the number of transposons always increases. But friends, when it comes to the cut and paste mechanism, friends, what did I tell to all of you? The number is basically getting decreased. Do not get confused with that particular point. Good afternoon, Apurva. Continue. Okay, we have understand clearly about the replicative and we also understand the non-replicative. And friends, I hope every student remember very clearly about an important gene. What do you call it, that particular important gene, friends? Transposase gene, is it? If you all people very quickly recall, every student remember my words. The transposase gene itself coding for the transposase enzyme. Friends, please never ever confuse in a given statement, because the statement were asked in this. I'll show you now. Now remember in your mind that transposase enzyme is not an external enzyme. Good afternoon, Akshay. Continue. The transposase enzyme is not an external enzyme, friends. The transposase gene only basically coding for this particular transposase protein or enzyme. And friends, the question is very clear, where exactly the transposase enzyme is going to cleave the DNA molecule. Every student remember that transposase gene exactly cleave the DNA at the terminal repeats. Where, friends, it will cleave exactly at the terminal repeats. Don't get confused with that. And friends, we have ended the last class by discussing this particular transposition. I hope every student remember very clearly. What did I tell you? In the grapes there are three colors are there: black color, green, or generally we can call it white, and another color is called as, friends, red color. And friends, if all of you quickly recall my statement, I have also given you one important statement. Just give me one moment. Yeah. So I have given you an important statement that, friends, there are the transposons are generally mutagenic. Friends, this statement is a really important statement. Never ever confused, because in order to make us confuse, they will give the statements very complicated statements. That is what transposition is generally mutagenic. Friends, it doesn't mean that the transposition always cause the mutation. That statement is a wrong statement, friends. What you have to remember is that 90% of the cases, friends, the transposition is mutated. There is no doubt about it. But what is the unique statement every student has to remember? In certain cases, the transposition is also beneficial. Not in a great amount, friends. 10% of the transposition will be beneficial. In that, we have studied one example, grape color by the transposition. I hope every student remember. I am not explaining you once again. I'll quickly summarize it. What is the name of the gene that is basically going to code for the anthocyanin? Friends, I want you to remember this particular name of the gene, VVMYBA1 gene. And what did I tell to you, friends? I told you that this particular gene is actually coding for an important pigment called as the anthocyanin. Please do not forget this particular statement. And here comes a very interesting point is what, whenever this particular retrotransposon, friends, remembering the names are very important. Here, what is the name of the retrotransposon? GRET1 is the name of the retrotransposon, will go and introduce near this particular VVMYBA1 gene, and it will disrupt the function of that particular gene. As a result, what happened, friends? Anthocyanin production never occurred. Whenever there is a no production of the anthocyanin, now, friends, what color of grapes we are getting, friends? Of course, we will get the green or white color. And what is the statement I told to all of you? We all people very clearly know that this transposable element never stick to one place, is it, friends? Now it's keep on moving from one place to the another place. Now every student remember, now this GRET1 transposon, when it come out from the from this particular gene, then most of the students has to think that it has to get back its anthocyanin gene, then once again it has to code it for the black only. But that is not happening. Why? The reason is very simple. Only friends, 90% of the anthocyanin gene is getting moved. 10% of the anthocyanin gene is left near this VYMYBA1 gene. As a result, friends, that particular gene is not actually coding for anthocyanin. So it is not giving rise to the black color, it is not giving rise to the white color, and it is giving rise to the third type of the grape, that is the red color. And friends, I have also given you one more very important statement. What is that important statement I have given? Whether it is the black color, white color, red color, friends, remember, some are more. They are having equal nutritional importance only. Please don't think that they are having different kinds of a nutritional importance. No, the nutritional value is some are less equal only. Don't get confused. What is the ultimate statement I would like to conclude here is that the transposition is the one which is actually responsible for the color of the grape. Very simple point it is. Now friends, I am going to explain you some more examples of the mutagenic effects of transposition. Friends, please listen. From this particular slide, every student remember, from this particular slide till the end of the PPT, if you listen me very correctly, you can answer the questions whichever asked in the previous year exams without reading only. Okay, without reading only you can answer the questions very easily. So I am requesting all of you understand the concept here. Just 5% of the concept you have to be remembered, friends, whereas 95% of the concept, friends, to be understood. That's why I'm requesting all of you understand each and every single line that what I am explaining. The moment you clearly understand, trust my words, you can answer the part C or part B, whatever the question that they ask in the PYQs, just like that. Now focus. I have given you two statements. What is the first statement I have given? The transposable elements are generally mutagenic. Most of the times it causes the mutation. But what is the another important point? Another important point is that transposable elements are also beneficial, but not all the time. They're just beneficial. Now remember, transposable elements are the powerful tools for the researcher. Now they can ask, they can give one of the statement. What is the statement that they can give? Transposable elements that plays a key role in the gene mapping. Transposable elements that plays a key role in the gene mapping. Then now they can ask, this statement is, the given statement is true or false. Every student remember, obviously it is a true statement. Now you can ask me, suppose it is the gene. Now this is the transposable element. Now the transposable element, it has moved from this area to this area. Now friends, we can easily understand, what we can understand, at this particular area the gene is basically going to code for which protein. We can easily study the gene sequence, we can easily study the gene mapping. Next, if the transposable element moved from this area to here, it entered, now we can easily study what this particular gene is going to code for. So friends, to understand the gene mapping and to understand the mutations especially, please listen with patience, I want you to listen. To understand the mutations and to understand gene mapping, researchers have genetically engineered an important transposable element named as sleeping beauty. They can give one statement in the exam. What is the statement? Sleeping beauty transposable element plays a key role in the cancer research. What is the statement? Sleeping beauty transposable element plays a key role in the cancer research. If you really don't know about that sleeping beauty, most of the students will think that it is not at all a transposable element, and 99% people will give the wrong statement. Now remember, the first genetically engineered transposable element is the sleeping beauty. What we understand, this is really interesting statement. So it is the transposable element. What is the common statement? We all know that transposable element never ever inserted into the DNA at one particular place. We all know that very well. It always introduced randomly. But what most of the scientists understand, in 40 to 50% of the cases, every student remember this beautiful line, 40 to 50% of the cases, the transposable element named as sleeping beauty, it is getting introduced into the genes which are basically causing the cancer. Understand friends very clearly. See here, sleeping beauty to induce mutations in mice, and it is used to search. Every student remember, we are using this transposable element generally to search for the genes that cause the cancer. Now you have to get one more valid doubt. Sir, then by using sleeping beauty, if we really comes to know that a particular gene is causing the cancer, then we can prevent the cancer before only, we can diagnose the cancer before only. Friends, that statement is true. We can diagnose the cancers very easily, but we never know in which stage that particular cancers are there. And moreover, I never use the word that 100% the transposable element sleeping beauty will exactly recognize the genes that are causing the cancer. No, I never say that. What people understood that out of 100%, only 40 to 50% of the cases, the transposable element sleeping beauty recognizing the genes which are causing the cancer. But I never say that by using the sleeping beauty we can always detect the genes which are causing the cancer. That is wrong statement. Don't get confused with that particular point. Now friends, now I am going to explain you the mutagenic effects of the transposition. Now they can ask us a statement in a given 4-marker question. Please listen the lesson very carefully. By that you can answer the questions without reading. That's what my intention is. Now friends, they can give the statement that transposons generally causes deletions. Transposons generally causes inversions. Transposons generally causes the process of duplication. Every student, please remember friends, obviously the statements are absolutely true statements. The transposons cause duplication, deletion and inversion. You people no need to remember how they cause. What you people has to remember is that they cause the deletion, duplication and inversion. Anyway, for your better understanding, I will explain it briefly. But according for the exam point of view, remember the statements that transposons cause all these kind of mutations. Apurva, that's what I am saying, dear. Okay, look into the point, Apurva. What I said is, suppose Apurva, look into this diagram. This is see here, this is the gene 1, this is the gene 2. This, whomever did not understand, please focus. This is gene 4, this is gene 5, this is gene 6. Gene 1, gene 2, gene 3, gene 4, gene 5, gene 6. Okay. Now what is the word I am actually using is, see, this is the transposable element. Now understand Apurva, the gene 4 causing the cancer. Now when we introduced this particular transposable element in this particular DNA region, 40 to 50% of the cases, everyone listen, 40 to 50% of the cases, it is getting introduced in the gene 4 region. 40 to 50% of the cases. Then when the scientist observed the function of that particular gene, when the scientists study the nature of that particular gene, they understand that this gene is majorly involved in causing the cancer. See, understand Apurva, it doesn't mean that the transposable element is not introduced at the gene 2, the transposable element is not introduced at the gene 5, it doesn't mean that the transposable element is not introduced in the gene 6. It is introducing everywhere. It is introducing everywhere, but in 40 to 50% of the cases, it is introducing at one particular gene only. So when we study the function of that particular gene, see here, then we understand that it is involved in the tumor formation. That's why we have written a line. See this line, Apurva, occasionally we used. Occasionally, transposon inserted itself in the regions which led to the development of tumors. It is not specifically introducing in that particular region. The insertion is a random insertion, there is no doubt about it, but what we observed is occasionally it will introduce at that particular region only. You got that point? Let me know if you did not understand. I'll re-explain a different way. Okay, friends, listen, focus. It is involved in the duplication, deletion, as well as the insertion. That is what I said to all of you. All the students listen. How does it cause the deletion? How does it cause the inversion? How does it cause the duplication? I'll explain to all of you. This is the gene. You can see, right, friends, this is the DNA having different kinds of gene. A gene, B gene, and yellow color one is what, friends? Transposable element. C gene, D gene, E gene, transposable element, F and G. Okay, very good. Continue. Now, friends, every student, please listen my statements carefully. I am marking here, this is one transposable element and this is one transposable element. My friends, what is happening now? This transposable element starts crossing over. Every student, please remember that this transposable element is doing the crossing over to the another transposable element. Now you only look into the diagram very carefully. Gene A, gene B, transposable element, C gene, D gene, E gene, transposable element, F and G. Now every student pay attention. See here, now there is a crossing over. How does the crossing over occurring? Everyone focus, friends. Gene A, gene B, transposable element, gene C, gene D, gene E, transposable element. Then F and G. In one line, what I would like to say is, if the two transposable element wants to be crossing over, there should be a compulsory formation of a looping structure. What, friends? Listen my statement. In order to allow the two transposable elements to undergo the crossing over, there is a looping occur. Because of looping, what is happening? See here, friends, there will be a deletion occurring. What is deleting? E gene, D gene, one transposable element, and C gene is happening. C gene is getting deleted. Then what we are left with? A gene, B gene, transposable element, F and G. So why I am explaining this to you? I am just explaining to make you understand that transposon can cause the deletion. You people no need to remember how it is causing. What you have to remember is whether it caused the deletion or not. That is the important for the exam. Now, if you look into this second case, look into this, everyone. A, B, transposable element, C gene, D gene, E gene, transposable element, F gene and G gene. Now, friends, what is happening? Once again, there should be crossing over is occurring between the two transposable element. Because of the crossing over occur in the two transposable element, I will write here what is happened is inversion happen. A gene, B gene, transposable element, E gene, D gene, C gene, transposable element, F and G. Friends, what happened? Just there is a change in the position of the gene. A gene, B gene, transposable element, E, D, C, transposable element, F and G genes. Very simple. And remember that it can cause the inversion also, it can cause the deletion also. And every student, please keep it in your mind. Even it can cause the duplication also. Focus, everyone, without having any confusion. Try to understand. See, all the students listen, look into the diagram. There is a gene, B gene, transposable element, C gene, D gene, E gene, transposable element, F gene, G gene. Friends, without confusion, please understand this third diagram. There is another gene is there. Both are sister chromatids. Both are sister chromatids. So A gene is there, B gene, transposable element, C gene, D gene, E gene, transposable element, F gene, G gene. Friends, don't get confused, focus. Now what is happening, friends? You all know, right? Every student focus. A gene pair with A, B with B, C with C, D with E, D with D, E with E, F with F and G with G. That is how they have to crossing over. But every student remember, without confusion, everyone pay attention here. Originally what has to be happened, friends? These two transposable element has to be paired with each other, and friends, these two transposable element has to be paired with each other. But every student, please follow me correctly. You know how does the pairing occurred, friends? This transposable element paired with this, and this transposable element paired with this. As a result, one chromosome got deleted, friends, and one chromosome got duplicated. Now you only look into the diagram, you will get a very good idea. Don't confuse, it's easy, friends. See here, every student, gene A pair with gene A, gene B pair with gene B. Now my friends, here there is a transposable element, right? Every student remember, this is the transposable element. Now you only tell me, does this transposable element immediately paired with this? No, not at all. What happened? This transposable element will go and pair with this kind of transposable element. As a result, you can see, friends, there is a loop got formed here. Because of the loop, all the students, please listen to me carefully. Now what got deleted, friends? Every student remember, this entire thing got deleted. C, D, E, this got deleted. Everyone remember, C, D, E got deleted. Now what are left with? Left with A gene, B gene, transposable element, F gene and G gene. So what happened, friends? In one sister chromatid, deletion occur. Now you only look into the next sister chromatid, A gene, B gene, transposable element, C gene, D gene, E gene, and once again transposable element, C gene, D gene, E gene, then followed by transposable element, F gene and G gene. What's the statement you have to remember? Because of the transposition which is unequal. Once again remember, because of the unequal transposition, in one sister chromatid deletion occur, in another sister chromatid duplication occur. So what is the statement I would like to conclude? I would like to conclude a statement that because of the unequal crossing over, because of unequal crossing over between the transposable elements, it will cause the deletion, it will cause the inversion, and it will cause the duplication. That is the three statements I want you people to remember without fail. Okay, and friends, especially from this particular slide, okay, that is the slide called as regulation slide. From here only, friends, regulation and types, from here only most of the time we are getting the questions, either from the regulation part and the types. These are all the two ways where they are repeatedly asking the question. So for the next 10 slides, I want everyone to understand very correctly, and ask me very immediately if you don't understand. I'll make sure that you'll get a good concept. Regulation of transposition. Now every student listen to me. So this is a DNA, this is transposable element. Now the transposable element moving from this area to this area and from here to here. So we all know, just now we studied, friends, whenever the transposition occurring, deletion, duplication, inversion occur. As a result, what happens, friends? The gene lost its function. Yes or no? The gene lost its function. As a result, what is happening, friends? The cell may die, which leads to several genetic abnormalities. Now friends, every student remember, it is compulsory, we have to regulate it. Now in a given 4-marker question, they will give one statement. Is there any which of the following mechanism act as a regulatory mechanism for the transposition? Once again, listen, remember the question. Which of the following mechanism is a regulatory mechanism for the transposition? There are many mechanisms that can regulate the transposition, friends, but among all, the major one is the methylating. This is one of the major regulatory mechanism. Many, see here, every student understand, if there is a no mechanism to restrict the transposition, the number of transposable elements would be increased continuously. As a result, friends, the host DNA is getting harmed. Please don't get confused. And cells have evolved defense mechanisms to regulate the process called as transposition. What is that regulatory mechanism? I'll ask you in the tomorrow class. I want every one of you to remember. That is the methylating. Now you can ask me, sir, what happens when the methylation occurs? This is really a wonderful concept. You all people know that, friends, if the transposition wants to occur, what is the enzyme that we required? We require an important enzyme called as a transposase enzyme. Every student remember, friends, this is really good concept. Now, who is going to synthesize this transposase enzyme? Understand the depth of concept. The transposase enzyme can be coded by the transposase gene only. That is what we studied. So the transposase gene is giving rise to the transposase enzyme. Then the transposase enzyme will do the transposition. Nothing but moving the transposable element from one place to the another place. Now what is my goal? Now my goal is I have to stop the transposition. Simple point. How can I stop? Suppose example, if I block the transposase gene, every student remember, if I block the transposase gene, do you think transposase enzyme can be synthesized? No way. If there is a no transposase enzyme, do you think transposition will occur? No way. So very simple point is what? We have to block the transcription and translation of the transposase mRNA. Such a simple step it is. If you block the transposase mRNA not to undergo the transcription, not to undergo the translation, friends, there will be a no synthesis of the transposase enzyme. If there is a no synthesis of the transposase enzyme, no transposition at all. Especially, friends, please remember, this is also asked question, friends. Drosophila melanogaster, which is a fruit fly. Fruit fly, you know, friends, what is the technique used by the fruit fly? Fruit fly will use the method, we all know that, RNA interference. By using the RNA interference means what? We never allow the RNA not to get transcribed and not to get it translated. If it is not getting translated, there is no synthesis of protein. By using RNA interference technology, they will block the action of transposase gene. Whenever there is a blockage of transposase gene, no transposase enzyme. So you people simply remember 3 steps. First regulatory mechanism, just remember these 3 steps. That is the DNA methylation. First regulatory mechanism. DNA methylation suppress the transcription. Then the second one, RNA interference. Remember as it is. And the third one, blocking translation. Okay, these are all the 3 methods by which we can regulate the transposable elements, or we can regulate the movement of transposable element from one area to the another area, which we can call it transposition. Simple point. Now friends, please listen. As I told you, the upcoming slides are extremely important slides, friends. Please pay attention. Transposable elements in bacteria. Friends, every student remember, bacteria contain 2 DNA. Bacteria contain what I told you, friends. When we human beings, we contain both DNA and RNA, but in our body, RNA transposons are more common, more predominant. But friends, when you look into this particular DNA transposons in the bacteria, see here, there are 2 types are there. Friends, both types are extremely important types. I am requesting all of you pay attention. Don't get confused. I'll explain it in a simple way. The 2 type of transposable element. First one we can call simple transposable element, and second one we can call, friends, composite transposable element. I can tell you in a simple word. Simple transposable element, which means it do not contain any external information. which is required for the bacteria. Once again, everyone listen my statement. Simple transposable elements are those type of transposable element. They do not contain any extra information required for the bacteria. What do you mean by that? So what does it contain? That is an important point. Simple transposable element contain the information which is only required for the movement. Every one of you please look into the diagram very carefully, friends. You only listen. What is this whichever I am marking with red color? This is the transposase gene. We all people know that transposase gene can code for what enzyme? Transposase enzyme. And you can see this green color one. What are that, friends? Inverted repeats. And what is this particular pink color one? We all know that flanking direct repeats. Now you only tell me, does it contain any extra information? Not at all. That's why we use the word simple transposable element. It do not contain any extra information. All the students remember, transposable element contain a transposase gene, flanking direct repeats and inverted repeats. Transposase gene can code for the transposase enzyme. With the help of the enzyme only, transposable element will move. It did not contain anything. Now everyone remember, it is coding for which enzyme? Transposase enzyme, correct na? Those sequences only we can call IS1, IS2, IS4 and IS5. These are all the sequences which can code for the transposase enzyme. Now let me simply define you composite transposable element. Friends, use the brain. So composite transposable element contains what? You can tell me very easily, it also contains transposase gene, no doubt about it. And it also contain inverted repeats, no doubt about it. It also contain flanking direct repeats, no doubt about it. But it also contains certain extra genes which will be useful for the bacteria. Suppose an example. There is a name of the transposon. Name of the transposon which is present in the bacteria, my friends. TN10. Every student remember, what does it contain? Let me tell you, it contain transposase gene, it contain inverted repeats, it contain flanking direct repeats. What else it contain? It also contain a tetracycline gene which is helping the bacteria to become resistant when the bacteria was kept under tetracycline medium. In one simple line, I will differentiate. Composite transposable element basically contain an extra information or extra information which is required for the bacterial growth and development. So for that example, we have taken the tetracycline. So what is the name of that transposable element? TN10. When it comes to the simple transposable element present in the bacteria, they do not contain any extra information required for the bacterial survival, growth and development. Simple transposable element only contain the information which is required for the transposable element to move. That's all. We can get a part B question also as well as the part C. I'll show you now. Friends, right now we have understand the transposable elements which are present in the bacteria. Simple, composite. I want you to remember nothing else. Okay. Now the transposable elements which are present in the phase, bacteriophase. Listen, you have to answer the questions. In case of a mu phase genome, the transposable element contains certain extra genes. You know, friends, what kind of extra genes they contain? The transposable element contain the genes which are required for the head, tail of the bacteriophase. So all the students look into the diagram, very simple. So this is the mu phase genome. Mu phase is the example of the phase. In this mu phase, there is a presence of the transposable element. There is a inverted repeats. There is a flanking direct repeats. Apart from that, The mu phase transposable element also contain, you can see here, head and tail genes and some other genes which are required for the development and growth of that particular phase. So what I mean to say, I can use the word, friends, now in the phase, whatever the transposable element is present, they are what kind of transposable element? Undoubtedly, friends, they are the composite transposable element. There is no doubt about it, because it contains transposase gene, flanking direct repeats, inverted repeats, and it contains an extra sequences which are required for the growth and development of that particular phase, like head and tail genes. That's why you can remember the statement in the phase the composite transposable elements are present. Okay, now friends, which is the most toughest one among all? That is transposable elements present in the eukaryotes. Okay, this is only most difficult one. Okay, if you get a difficult listen my words, don't worry. If you get a difficult experimental based question, if you get a difficult experimental based question, you will get only from the eukaryotes. Okay, so I'm requesting all of it, is too confusing and too tricky. I'll try my best to make it extremely simple, and I want you to focus and understand. If you understand very clearly, 4 marks in our pocket. Okay, focus. And friends, so in case of eukaryotes, focus friends, DNA transposons are there and RNA transposons are also there. And I also told to all of you friends, remembering the names is extremely important. Never ever forget the names. Example of the DNA transposon, P elements present in the Drosophila. P element is the name of the transposon, never forget. Okay, AC and DS is the name of the transposable element present in the maize. And these are all what, friends? DNA transposon. Don't forget, don't forget. P elements are the type of transposon present in the Drosophila. AC and DS elements are the type of DNA transposons present in the corn. Example of RNA transposon. TY element is the name of the transposon present in the yeast. The most important one, Copia element is the name of the transposon present in the Drosophila. Don't forget, friends. ALU sequences which are present in the humans. We read now, friends, ALU sequence, sign and line. Sign and line. Short interspersed nucleotide sequences. Just remember short names. Sign and line. Which are the DNA transposons, which are the RNA transposons, where they are present. That is more important, friends. Now, majorly we are studying about the AC and DS elements in the maize. Please listen, you have to answer the questions. Barbara McClinton, she is the woman scientist, who is really a brave woman. Okay, and I don't know whether you have studied her story or not. She is the first scientist who refused to take the Nobel Prize. But later she was awarded. Please read her story, it is really inspiring. Barbara McClinton, she was the first one who identified the transposable element present in the maize. Friends, what is the name of the transposable element present in the maize? AC and DS. Please remember, what is in one line, what is her exact contribution? Her study was on maize genes that caused variegated kernel. Friends, we all eat corn. Correct na? You know, right? So we will eat a thing like this. What is this called as kernel, right? We all know, right? Kernel originally which in color? We all know that it is yellow in color, correct na? And if it is a very young, then what we can, it will be in a white color. Only 2 colors you can observe, yellow and white. You might observe many times when you are eating this particular entire corn, you might observe very clearly that some are brown color. Then we will just remove it, we will throw it and we will eat the remaining one. So that remaining brown color and slight reddish color are there. What do we call them as? Variegated kernel. What do you mean by the word? Listen friends, listen to me for 10 minutes of focus. Variegated means multicolored kernel. It's really interesting topic. Why I used the word multicolored? Multicolor is caused just because of the transposition. Let me explain you how. Okay, focus friends, don't get confused. It's a simple topic. Transposable elements in eukaryotes. Friends, remember, all the students look into the diagram. This is AC element and this is the DS elements. These two are the examples of transposable element present in the maize. Now friends, all the students remember, if you look into the structure, friends, everyone please pay attention. Look into the structure. This is the transposase gene. We all people know that. And this is the green color one, what friends? Inverted repeats. Pay attention. And this pink color one is what friends? Flanking direct repeats. No problem at all. Now every student has to remember my statements very carefully. The AC element, which is a transposable element, it contain a gene. What gene it contain? It contain transposase gene. Now we all people know very well, it contain the transposase gene. Undoubtedly it contain the transposase enzyme, no doubt about it. So the enzyme will cut at the inverted repeats. Then this transposable element can move very easily, very easily. The AC element can move. Why I am using the word AC element can move? Because AC element contain transposase gene which can code for the transposase enzyme which will cut at the terminal repeat, so it can move. But my friends, all the students remember, that's why we can call, everyone remember please, the transposable AC element are called as autonomous. Why do we use the word autonomous? They can move on their own. Because they contain enzyme, they can move, no doubt about it. But every student look into this. This is the DS element. DS element is also name of the transposon only. There are many DS elements are there. DS9, DS2D1, DS2D2, DS6. There are many transposable elements are there which belongs to DS. And every student look into the diagram. This is what gene friends? Transposase gene. If you look into the 4 diagrams, everywhere the transposase gene got deleted. The transposase gene got deleted. Now what is the statement every student has to remember? Now the DS elements are not autonomous. Then what they are? They are non-autonomous. What do you mean by that? Which means friends, they cannot transpose on their own. Why they cannot transpose on their own? Because if they want to transpose, there will be a compulsory requirement of the transposase enzyme. That enzyme is coded by a gene called as transposase gene. That gene is absent, or sorry, that gene is deleted in the DS. That's why they are non-autonomous. Now you have to ask me a very valid question. Sir, here you said that the AC transposable element is moving on their own. There is no doubt about it because it contains transposase enzyme. Now my question is, how does the DS transposable element is moving? Because it do not contain the transposase enzyme. That is the question. But here comes the answer. The DS transposable element also can move. There is no doubt about it. But how? It can move in the presence of a transposase enzyme of AC element. Very simple. Don't confuse. You all listen to me. Everyone listen friends. I am AC transposable element. You all people are DS transposable element. I am AC transposable element. I am having my own enzyme. So I can move easily. And you all people DS transposable element, you don't have any enzyme. You can't move. But if you want to move, you have to take my help. So in the presence of my enzyme, I am also having a transposase enzyme. In the presence of my enzyme, you all can move. Because you are all DS elements, you can't move on your own, but you can take my help and you can move. So that point you have to remember. That is what written here. That is what makes us confused. AC elements are autonomous, able to transpose on their own. Myself. DS are non-autonomous, cannot transposes on their own. You people. but you people can, which are DS elements, can move in the presence of AC elements. Such a simple step it is. Now if you understand the upcoming 3 slides, the concept is crystal clear, but it is too confused concept. Pay attention, use the brain. Now we are talking about the AC and DS transposable elements. What are their function, how they are coding. Don't get confused, just use the brain, apply the knowledge, you'll get a very good idea. Focus friends, everyone. A pigment encoding gene in a corn kernel is determined by a capital C and small c. So friends, capital C and small c, which is a heterozygous, it is the one which is representing the color. Every student remember, if you take a kernel which is homozygous recessive, every student remember, if it is a homozygous recessive, it is yellow or white. If it is either homozygous dominant or heterozygous, which is the purple. Friends, if you don't remember this, you can't understand the concept at all. Every student remember, color can be given by this particular loci, capital C, small c. If both are homozygous recessive with our 2 small c, the kernel will be yellow color or white color. If it is a homozygous dominant or heterozygous, it is going to give rise to the purple color. Now every student look into the picture. Now you have to understand, please use the brain. What happens if the transposition is not occurring? What happens when the transposition is occurring? If you use the brain, you understand. If you try to buy hard, you never get it. Use the brain and listen to me. And this is the first case. What is the first case? Transposition is not occurred. All the student listen. This is the AC transposable element. This is the DS transposable element. Now friends, what is this particular phenotype here? Small c and small c. Then you only tell me what will be the phenotype. The phenotype will be yellow kernel or the white kernel. You understand it very clearly. Now this is easy to understand. What every student has to understand without confusion, there will be a no transposition here also. Now see here, AC transposable element, DS transposable element. Now here every student remember, here it is the capital C and it is the small c. Capital C, small c, nothing but heterozygous. And what we will get? We get the color of purple. Now once again remember, 2 small c's, yellow kernel or white. Capital C, small c or homozygous dominant will be the purple. Now you got it clearly. But what is the condition in 2 cases? In the 2 cases, transposition is not occurred. This is easy to understand. Now you have to understand very clearly what happens when the transposition occurs. If you understand this very clearly, the concept is clear. Focus. This is too confused slide. Okay. Focus everyone. Friends, I will repeat it twice or thrice. Okay. Apply your brain and understand. This is the condition where the transposition occur. Okay. This is the AC transposable element. And friends, this is the DS transposable element. Every student has to listen to me. And this is the gene which is coding for the capital C. Capital C. Capital C, it is basically going to code for the purple. Now, you all people listen to me carefully. Anyway, you only tell me, does the DS transposable element can move on its own? It's impossible. It cannot move on its own. But what is the unique statement? The DS transposable element can move in the presence of AC. Every student has to listen to me. Now the DS transposable element in the presence of AC transposase enzyme, it entered here. Now you only tell me where it entered. It entered into the gene which is going to code for the purple. Now you have to answer my questions. Now what the DS transposable element is doing? It is blocking the function of this gene. Now it blocks the function of this gene. Do you think the gene can code for purple color? Now you all the students look into this. Transposable element entered here, the DS1, and it disrupt the function of the gene. Hence friends, you can't get the purple. What you get? You can get the yellow color. Use the brain, understand, it's easy. Now the second condition, I'll re-explain, don't worry. Same. Now the question is very clear. Now anyway, same picture. AC transposable element helped the DS transposable element to enter into the area where it is going to code for the purple. The same case, same case. Just listen, just listen, just listen, just listen. Now what happens? We all studied very clearly that friends, transposable element cannot withstand at one place. It's keep on moving, keep on moving, keep on moving. Now you have to, all people has to answer my question. Now the DS transposable element moving from here to somewhere else. Now you all has to answer, once I remove the DS transposable element from the gene which is basically going to code for the purple color, it has gone. Now you have to understand that once again the gene regain its function. Now once the gene regain its function, sir, then it has to code for the purple. Yes or no? Of course yes. But see here, it is going to code for the variegated kernel. You have to listen to me. What do you mean by that? Out of 100%, only friends, 80% of the DS transposon got removed out. Remaining 20% of the DS transposon is left. So you only understand, wherever the 20% of the DS transposon is present, there it cannot code for color, hence it gives rise to white. Now wherever the transposon is present, DS transposon is present, it gives rise to the, wherever it is not present, it gives rise to the pink color. That is what we can call a variegated kernel. Don't worry at all, I will explain 2 more times. You will get a crystal clear idea. I want you to get it very correctly. Focus. AC, and you all can see this. This is the purple color gene, which is, this is the capital C gene, which is basically going to code for the purple color. Now the DS transposable element, in the presence of AC transposase enzyme, it entered into that particular gene and disrupted the function of that gene. Once it disrupts the function of that particular gene, it never ever code for the purple. It is going to code for the yellow or white. There is no doubt you got that. Why we get the white color or yellow color? Because the gene got disrupted. Now we all know that this transposable element which is entered into the gene, it never present in that particular gene for the long period of time. It's keep on moving. So when this DS transposable element, it has moved from this particular gene. Now what the question every student has to get, once it move from this particular gene, then we have to get a complete purple color kernel. Why we did not get? The reason, entire 100% of transposable element is not left. Only 80% is left, 20% is remaining. Wherever the transposable element is left, there we will get the white color. Wherever the transposable element is not there, we will get the purple color. So in one kernel only, we are getting white color and purple color. That's why we can call a variegated kernel. Variegated means what I said to all of you, multicolor. Okay. No. Apurva, that's a wonderful doubt. AC is not moving, dear. Only the transposable element DS is moving. But the question is here, the transposable element DS is moving with the help of AC. That's the point. Both are not moving. Okay. Friends, everyone please respond. Is it clear or not without having any confusion? I strongly believe that everyone understand without confusion. Please respond. Say yes or no. I will re-explain if anyone feel any kind of difficulty. Okay. Now let's see whether you learned it correctly or not. Let's try to solve the given questions. See friends, I already told you many times, reading, revising, listening, studying, everything is different when it comes to answering a question. When it comes to answering a question, everything is different. Okay, use your brain, do not confuse, slowly answer the questions. Okay, here you go. Okay, this is the question which is asked in your 2022 February question paper. in the part C. Don't be hurry. See, such a simple question they have asked in the exam. Slowly, slowly, slowly. Slowly, Neha. Just now I told to all of you, dear. Slowly, slow, slow, slow. Okay, good, 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 good. Simple. Very good. The answer is D. Very simple, straightforward, part C question. Okay. Now, don't be hurry. Think and answer this question. This is 2020 November part C question. Good, 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 good. That question is very simple question. And think and answer. Okay, what is the answer for this question? Even though you know the answer, recheck. Don't be hurry. Okay, slow, slow, Sumaya, slow. I told you many times not to get excited. Slow. Don't give me the answer before 1 minute, please. Sumaya, see dear, the simple question you made it wrong, huh? See here, B and D. Okay, you're saying like that. Huh? Look into the option correctly. Okay, okay. It's very simple, right? We have very clear, slowly. We have very clearly studied that eukaryotes contain DNA and RNA, but RNA is the predominant. When it comes to the prokaryotes, what did I say to all of you? Prokaryotes contain only DNA. Correct, na? Most mobile genetic elements in the bacteria transposes via an RNA intermediate. No, most mobile genetic elements in the bacteria are DNA. In case of here, not retrotransposons. Both RNA and DNA transposons are found in eukaryotes. Hence, the correct answer will be, friends, B and D. Okay, very, 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 very simple question asked for 4 marker. Okay, so these are all the questions. So especially when you are reading the transposon, first remember the types, then remember the mutations, and remember the genes. Okay, apart from these 3, there is no way of asking the question. Okay, I'm requesting all of you revise this thoroughly. Hope it's clear for everyone. Hope the discussion is useful to all of you. We'll meet in the tomorrow. Friends, only for tomorrow, I'm requesting all of you only for tomorrow. Please attend the class at 10 a.m. Okay, only for tomorrow. Okay, so tomorrow we will continue the microbial physiology chapter. Is this question? Okay. A group of transposable element described as retro elements encompass. So they just asked which are present in the Drosophila and which are present in the humans. Okay, so here Anyway, lines and signs, you can easily eliminate option A and option B, Sumaya, very easily, because lines and signs both are present in the humans. Which one? Lines and signs. Okay, so hence you can see the option D. P elements are present abundantly in the Drosophila. But you know, it's a kind of a good option. P elements present in the Drosophila, copia elements are also present in the Drosophila. But the point is here, P elements are predominant, most abundantly present in the Drosophila. P elements. P element is a type of transposable element abundantly present in the Drosophila. Line and signs, which are a type of ALU sequences, which are present in the human being. The answer will be D. Got it correctly? Okay, revise once. So tomorrow we will meet at sharp 10 a.m. We will be starting the microbial physiology, the third topic from the cell biology chapter. Okay. Okay friends, thank you all of you. I really appreciate all your patience. Thank you so much. I'll see you in the tomorrow class. You understand, Sumaya, the question? Okay, good, great. I'll see you in the next class. Bye-bye. Thank you all.